Hey guys, welcome back. So we're going to move on to part 2 of Unit 1, which is looking at 1.3 and 1.4 from the AP spec. Macromolecules and carbohydrates. Once again, there's a share button. If you want to share it with a friend, also you can download this. It's going to open up and it's a static web page. Something I forgot to show you last time is I've got this little dark and light feature. So depending on your taste, I think the animations look quite cool in dark. Of course, they look great in light as well, but that's just there for you in case. Quality of life. It's the little touches sometimes. Now, we're going to look at macromolecules and in a bit more detail, specifically carbohydrates. So we've got some general language that we can use. We can describe something as a monomer. So there's monomers. Well, there's no true monomers in lipids. However, we do have these would be called fatty acids, what they've been broken up into here. And carbohydrates, a monosaccharide. So here's 3 monosaccharides joined together. The bond that would join it in pink is called glycosidic. And these are carbohydrates or sugars. Protein, amino acids are our monomers. And the bond between them here, the different amino acids, is a peptide bond. Now, in order to join 2 monomers together, we would have what's called a condensation or also dehydration reaction. So typically in AP, we're using the word dehydration to describe these reactions, but it's also worth knowing that they can be referred to as condensation. So it's the removal of water. It's also could be called a anabolic reaction. So the way that I remember that we're building something up is the mnemonic, or this, it's not mnemonic, it's just a silly thing, Anna the Builder and breakdancing cat. So anabolic is building something up, Anna the Builder, and breaking something down, which is addition of water, is going to be a catabolic reaction. So together, anabolic and catabolic reactions make up metabolism. The web of different reactions, which we'll get more and more into as the course goes on when we think about things like respiration and photosynthesis. Of course, all of these reactions are typically catalyzed by enzymes, lowering their activation energy, allowing bonds to be broken or formed at a lower energy state. So a nucleic acid, so DNA and RNA are nucleic acids. And if you remember CHO, CHOM, CHOMP, they have nitrogen and phosphate in them as well as the basic 3. And these bonds between them here are called phosphodiester. Now, all of these bonds are covalent bonds between 2 non-metals. And our simple monomer is what we would call a nucleotide in this example here. Now, these are representations of fatty acids, supposed to be like a little, so here's our our carbon hydrogen chain, and then this our COOH, like a little head bit of it. But we don't have true monomers in lipids. We have the ones that we look at would be steroids, phospholipids, and triglycerides. And we'll talk about more when we look at lipids particularly. We're going to move on and think about just carbohydrates today. And if we number our carbohydrates, I'm going to zoom in on this. And if we look here, carbohydrates, we number our carbons within a sugar, not number our carbohydrates. Slip of the tongue there. So this is C6H12O6. You'll recognize that as glucose. Most of us know that from learning about photosynthesis when we've studied like lower level sciences. And what's interesting here is this is B1, 2, 3, 4, 5, and 6 carbons. That's important when we talk about bonds, like a 1,4 glycosidic bond is between the first carbon on one molecule and the 4th carbon on another molecule. Or we might have a 1,6 glycosidic bond, so from the 1st and the 6th. Now, we've got these OH groups. This should be sticking up. And no, it should be sticking down. Sorry, so it should be coming down here. There's one little mistake in this picture. But the rest is good. Now, this is an alpha glucose. How do I know it's an alpha glucose? If I tap on this over here is going to tell me that drawn below is the ring is an alpha glucose. brought above this OH group is a beta glucose. So if I flip this OH up here, it's beta. That's relevant because when you join alpha or beta glucoses together, then you're going to have a change in the growth structure of that, so the polymer or the polysaccharide. Now, the way I remember that this is alpha. Now, when we think about an alpha in a group or like a lion pride or even a group of humans, then the alpha is the one that takes charge, that is not in the lead, but is in charge of the group. And here, when it's alpha, we just have one OH group sticking up. And the rest of them, if this one's sticking down too, the rest of them are sticking down. I'll fix this. It's not going to be fixed in my video, but it's fixed in the interactive lesson that you will be able to access afterwards. The link will be below in the description. So alpha, one of them sticking up, and beta, you'd have the first and the third sticking up. So it's like an alpha of a group. Now, if you want, you can click on the different parts and get some information as you go. Why sugars dissolve is a nice little fact. The other half of the bond. One that's mentioned, the 1,4 glycosidic bond. So yeah, have a little look through that. Build it, and then break it. So let's build. You can see this little stake on the fourth. Removal of water. This would be a 1,4 glycosidic bond. And if we were to break it, we would add water. So that's going to be a hydrolysis reaction when we add water back, or catabolic. And if we just skip back, removal of water. So an OH is taken from one and a hydrogen is taken from the other. And removal of water is condensation, also called dehydration, synthesis. Now, the structure will dictate its function. And the structure is going to also be dependent upon if there's 1,4, 1,6 glycosidic bonds, if it's alpha or beta glucose. So starch is composed of amylose and amylopectin. They're both storage molecules that comprise starch, and glycogen is also a storage molecule in animals. So starch, so here's our amylose, it's a 1,4 alpha glucose bonds, and it makes a spiral. Now, a spiral is okay for storing. Look, we got lots there, and it's compact. But what's better than a spiral for when we want to access this energy is something that's branching. So by having a 1,6 in there as well, we end up with these branches. So 1,4 and 1,6 glycosidic bonds means that we end up with branching. So this is 1,4 plus 1,6. And then cellulose just has straight chains. So it's 1,4 bonds, and these arrows here are, every other monomer is flipped. So if you can imagine that group that we see here at the top, the CH2OH group on the 6th carbon, that flips. And because it flips, we end up with a straight chain. And then here you will actually find weak hydrogen bonds, not any different than a normal hydrogen bond. All hydrogen bonds are kind of weak compared to like covalent or ionic, but you'll find these hydrogen bonds between the different chains. And overall, we've got a very strong structure that is cellulose, which is structural, and we all know that you find that in the cell wall. You would have noticed when I've been talking of things like a monosaccharide, a disaccharide, a polymer, polysaccharide, but that saccharide means sugar. And when we say about cellulose, OSE, glucose, cellulose, fructose, OSE is also another indicator that we're dealing with a sugar. ASE, maybe some of you already know, is for an enzyme. Now, how do we fit this all together? Now, monomers join to form polymers, and they're built up by condensation reactions. And then condensation and hydrolysis are opposite reactions of each other. Let's zoom in a bit more. And hydrolysis will break down polymers to take us back to monomers. Condensation links things together. Alpha and beta glucose, same formula, but they are what we call, and I didn't use this word before, an isomer. So same chemical formula, different structure. And that's just that flipping. Remember, alpha is the one where it only has one OH group up, and beta will have 2 up, the first and the third. Then, as before, I have some fill in the gaps for you to test how well you've been listening. So let's do the first one. Large biological molecules are polymers assembled from repeating monomers. And at the end, you can hit check and you'll see if you're right. And then I've got a few multiple choice questions. So a polysaccharide made of 60 glucose units is completely hydrolyzed to monosaccharides. How many water molecules are used? Now, if there's 60, or even like say if there's 2, we have 1 bond. If there's 3, 1, 2, 3, you're going to have 2 bonds joining together. So this is like a random question that they often ask in different forms, like how many peptide bonds are there between 300 amino acids? It's whatever the number is, minus 1. So feel free to have a go at these 6 questions. I won't do them all now. I want you to be able to have some for practice yourself. And don't forget that also a free resource that we have available under the multiple choice questions. When we look at AP, you will find some macromolecules and carbohydrates. And you feel free to share that as well. Teachers watching, you can actually share the link with your class. You can download it. There's a download button. And you can print it off too. So this will give you instant feedback. After a starchy meal, starch in the gut is converted into individual glucose molecules. Which statement describes this process? So hydrolysis, breaking stuff down, water is added to break covalent bonds. Okay, we'll leave it there for today. Remember, there's also, if you want help memorizing the basic definitions and also testing yourself on the applications, then I have my free study app, which is Cramly. Screen I was listening at before on here, under topics, definitions, applications. Why does sweating cool you down? You can get an adaptive algorithm that will grow as you learn. Lots of functionality. You can find it in the Play Store. It's completely free. It will never be paywalled. It's my gift to you. So, hope you enjoyed today's. Catch you next time, and ciao for now.